After last lecture, some of you asked me amino acids, right, the last few slides, how to approach the last few slides. I went rather quickly, right? So the idea is to introduce to you how we can use the carbon atoms of the amino acids, right? So we talk about degradation of amino acids, and we remove the nitrogen atom, the nitrogen moiety from the amino acids, and what we are left with is the carbon skeleton, right? So this last few slides is to tell you how we can harvest the remaining energy, or how do we use the products of the alpha keto acids. Right, so you see a lot of amino acids here, right? And some of you are really concerned and asking you to memorize all these things, which belongs to what and blah blah. I'll leave it to you to decide. But what I'm trying to convey here is that whatever is left from ammonia extraction from the amino acids, we use it usefully. And because of the way they enter the Krebs cycle to be harvested, right, for the energy, we have a new classification of amino acids that you have not seen before, which is ketogenic and glucogenic amino acids. Right, so the ketogenic amino acids are capable of producing energy via ketone body formation. Okay, we know that some of these organs use ketone bodies, right, for complete oxidation, and some of them, right, are converted to glucose and glycogen via gluconeogenesis. So indirectly contributing to TCA cycle, because the glucose will also enter the citric acid cycle. And out of these, you have 5 that are both ketogenic and glucogenic. Right? Okay? Right, so I think it's easier if you just remember the 5 that is less distinct, entered both, okay, can enter both the cycle as both ketogenic and glucogenic. And then we went on to talk about the classification that you know, okay, nutritionally you have essential and non-essential amino acids. What we mean by that, okay, essential amino acids because you cannot synthesize them. Okay, other organisms like plants and bacteria can synthesize all the 20 amino acids. We cannot. We can only synthesize half of it, right? And so we have 10 essential which we must take in our diet. Okay, we must take in our diet. And this slide shows you, gives you 3 examples of how we can make 10 or 20. 20 in this case for plants and bacteria, but for us is 10, using the precursors. Okay, so precursors, so meaning from glutamate we can get glutamine. We don't start glutamine from scratch. We build glutamine from glutamate, right? And glutamate we can have it here from the intermediate from the citric acid cycle, alpha ketoglutarate. And this reaction you know very well now. This is an alpha keto acid, this is an amino acid. So it is a transamination process. So is this, a transamination process. Okay, so the concept, right, the concept of transamination comes in. Okay, right? So as long as you remember the structure, you can tell the difference between the structure, okay, between keto acid and amino acid. Then you can link it up to this whole concept, transamination. Right? Even this is also transamination. You see, they are all, this is reductive amination, sorry, this is transamination. And serine you haven't seen from 3-phosphoglycerate. You can produce cysteine because they have the sulfur group in them. So you group all the sulfur group ones together. Serine, serine, not serine, serine has OH. Cysteine and methionine. Okay? Alright? Okay, so this is to tell you how we can make, build the 10 non-essential acids, okay, from precursors. Not everything is from scratch. Okay, so that's the idea that we're trying to convey in here. I'm not going to ask you in an MCQ question, is serine ketogenic or glucogenic? No, I'm not going to ask that kind of question. Right? Okay, now today we are at our last lecture on nucleotide biosynthesis the approach is slightly different from here. Okay, so you see that for glycolysis and TCA cycle, we're quite intense. We're intense on the enzymes, we're intense on the structures. Structures you have to remember. Okay, I've already said this. And then beta oxidation on fatty acids is the concept of moving the bonds. Okay, so not about remembering structures, right? And the enzymes involved, the special enzymes, the hydratase, the enolates, etc. Okay, and then the problems that you encounter when the bonds are in the wrong positions. Amino acids is about urea cycle, again removal of nitrogen. And the nucleic acids, how we're going to approach this is to see how we can build the molecule from precursors. So the focus is quite low and not so intense on enzymes and regulation over here. Alright, so you will see in your notes stepwise addition of precursor molecules on how to build finally the purines and pyrimidines. Okay, and finally the salvage pathways. What do we mean by salvage pathways? How do we salvage our base? Why do we salvage? Alright, and then lastly, the mechanism of action of 5-fluorouracil as an analog of your pyrimidine to inhibit the synthesis process. Okay? Alright, so the important roles of nucleotides, right, not so much in the case of diet anymore. Diet is glucose, amino acids, and fatty acids. In the nucleotides, we know that it's very important, right, because they are precursors of DNA and RNA. The genetic material that is primary, yeah, and we have also seen the high energy phosphates, the ATP and the ADP being used in our metabolism process, right? So adenosine triphosphate, yeah, and these are also the precursor molecules for polymerization of the DNA replication, right? And also the transcription processes, right? Even the cofactors, the nicotinamide adenine diphosphate. FAD and ADP, coenzyme A, they also have this nucleotide structure. Right? So how do we classify nucleotide? Right? How do we classify nucleotide? Do you remember? So we have three base, we have nucleoside, and we have nucleotide, right? Okay, this goes back to your Dr. Ho's lecture. So I hope we don't have any confusion. Okay, so let's say we have adenine, right? We have adenine here. What do we call this? This is our what do we call this? This is our free base. This is our base, right? Alright? And then we keep on building it. And we add on we add on ribose sugar. Okay, so we have a free base, and when we add on the ribose sugar, this is adenine. Right? And then we add on the ribose sugar, what do we call it? Adenosine. Right? And what is this? This is a nucleoside, correct? Nucleoside. Okay? Yeah? And finally, when we add on the phosphates, okay, you can have 3 of these, okay, and then this one will become ATP, isn't it? Adenosine triphosphate, and this is your nucleotide. Okay? Right? So no confusion about it. We have to be clear about this definition. Base plus sugar, nucleoside, and then base plus sugar plus phosphate, nucleotide. Yeah? Okay? So today we're going to look at the synthesis of nucleotide. Okay? Right, because you will find out that for purines and pyrimidines biosynthesis, they are not formed as a free base. In fact, they are formed as a nucleotide. okay, in the body, right? Straight away as a nucleotide, never as a free base itself, right? Okay, we'll look at the process today, and these are our cofactors. So you have variations. You have vitamins added, vitamins moiety added to the nucleotides, and you get all your cofactors, right? You have also other components like UDP, glucose. All these you have not seen, but they are participate in the metabolic process in gluconeogenesis. And this one we have mentioned just on the last slide, making cysteine from methionine. This is involved. So they are reactive intermediates. And we also have all these acting as allosteric effectors. They are not just substrates, but they are also allosteric effectors. And finally, second messengers you will see in pharmacology. Right? Why second messengers? So sometimes you have first messengers. The first messengers are your hormones, right? And your hormones usually are hydrophilic. They cannot cross the membrane, right, and go into the cell to transmit the signal. So they sit on the receptors on the cell surface, and they trigger your second messengers inside the cytosol to relay the message. And therefore we call it second messengers. And your second messengers are all nucleotide in nature, much smaller, right? And they transmit the message to the pathways inside the cytosol. So these important functions, right? Yeah, these are all important functions in the body. There are two ways whereby purines and pyrimidines. So purines, which are the purines? Do you remember your purines? What are your purines? We have A, T, C, G. So your purines are A and G, correct. Pyrimidines, C and T. Okay, AG purines, yeah, CT pyrimidines. So we can make them in two ways. The first way is called de novo pathway. That means you build it from the scratch, from new, completely from their metabolic precursors. And you will see that we use ingredients like amino acids, ribose 5-phosphate, carbon dioxide, and ammonia. All right. Okay. So this would amino acids will give us the carbon skeleton of the nucleotides. Ribose 5-phosphate will give us the sugar. Carbon dioxide will also give us contribute the carbon skeleton, and the ammonia will give us some of the amines, right, inside the purine pyrimidine structure. Because we won't have it, okay? We won't have much of it in the new amino acids. So this is from scratch, from precursors. But in the body, we also have salvage pathway, what we call salvage pathway. That means when nucleic acid breaks down, okay, the body again doesn't waste any materials. You can recycle the free bases or the nucleosides, right? If it's free base and you want to make nucleotide, then you have to add sugar and the phosphate on top of it, right? Then nucleoside, you have to add the phosphate group on top of it. So these are what we call salvage pathway. When materials break down, you harvest what is half broken, partially broken, and then you build it up to nucleotides again, right? You don't start from scratch. Okay? So we have two classifications, purine, pyrimidine, right? So this is your purine skeleton. This is the scaffold. Remember your purine, right? Your purine A and G, adenine and guanine, right? Okay. So over here, we have, this is the main skeleton. So your purines will have a 2-ring structure, a 6 and a 5. Yeah, your pyrimidine only has one 6-membered ring. Alright? So over here, as I mentioned, the nucleotide or the purine synthesis is made as a nucleotide, not as a free base. And the interesting thing is that we know that the purines is A and G, but the precursor is IMP, which is inosine monophosphate. That means this whole build-up process will not give us AMP or GMP directly, but it gives us a precursor called IMP. Right? So we'll see how we can get A and G from I. Yeah? Okay? So this IMP is synthesized through a purine base on the ribose 5-phosphate. Right, and after IMP is produced, right, you will derive AMP and GMP, and kinases will then add the extra phosphates onto it to give ATP and GTP. Right? So the way they figured out, it was in 1948 that scientists kind of figured out each of the components where it came from. So what they did was to feed raw materials to birds. Right? They labeled the raw materials with radioactive carbon, nitrogen atoms. Right? And then the last time we talked about urea cycle, we said that in human we remove our nitrogen as urea, correct? But then birds, pigeons, remove all their excess nitrogen as uric acid. Right? So by looking at the uric acid excreted by birds, they managed to figure out where these components come from. Okay, so we see that we can account for each of the atom on the skeleton. The first nitrogen, right, is from the aspartate amino acid. Right, so the aspartate involved in purine synthesis, you will see is aspartate, glycine, and glutamine. Right, so aspartate and glutamine, remember that they are nitrogen containing in the R groups, right? So it's very useful for building the purine rings, right? And then we have glycine. Glycine is the simplest amino acid we have. So aspartate contributes to 1 here. The numbering, right? I think you have probably learned in either in the earlier lectures or in MedChem that this is the way we number the urine ring, right? We start from nitrogen, we go anti-clockwise, then we cross over. Okay? That's why it's 1, 2, 3, 4, 5, 6, then we go to the imidazole ring, which is 7, 8, 9. Okay, it's quite a unique way of numbering. It goes this way, yeah? So the first nitrogen is from aspartate, right? And you then see that there's some kind of symmetry over here. The carbon atoms here and here is contributed by what we call a formate, or more specifically an N10 formyl tetrahydrofolate. Okay, this is the precursor. I will show you the structure and the donation. Okay, formate. Formate actually means CHO. Okay, formaldehyde, right? Formaldehyde is COHH. So you know your aldehyde group is this way, right? Your aldehyde. This is the aldehyde, right? Okay. So your formaldehyde has the R, which is an H. Simplest. It's the simplest aldehyde. Okay. Formaldehyde. Okay. So what it basically means is that they donate this aldehyde. Okay. This carbonyl in the form of aldehyde into the structure. Okay, so formate or formyl. Okay, formyl is this functional group, COH. Okay, COH group. Okay, so this COH group is donated by this precursor called N10 formyl tetrahydrofolate on both sides of it. Okay, and then at the bottom here, nitrogen. Okay, this nitrogen is donated by the glutamine, amino acid glutamine. And finally, you have this amino acid glycine. The whole backbone of the glycine is added. Right? Whole backbone of glycine is added into this purine ring. Okay, over here. And the last carbon here is contributed by a bicarbonate. Alright, by a bicarbonate. So our stepwise reactions will show us, okay, how we can build this ring like Lego. Okay, patch it up. This is the overall reaction. I put it here because my slides are all chopped down. So this will give you an overview how it is going to happen. Don't worry about the colored pictures here. These are just the shapes, the ribbon structures of the enzymes involved. We are not interested in that. Okay, but I couldn't get rid of it completely. So if you see over here, the way that purine is being built, we have about 11 steps. Okay, so 11 steps. Let's go through it now. Yeah? So, key concept. Okay, at the end we have precursors molecule, we have amino acids, we have bicarb, we have ribose 5-phosphate. Did I miss out anything? And formyl, correct? Formyl. These are precursors, okay, to give us our purine. And finally, the end product is IMP. It's not A or G directly, it's IMP first. So let's see how we can get to IMP. Okay? So back to this, IMP looks like this. Okay? So it's inosine monophosphate IMP. How does A and G differ? A is, instead of a CO over here, it's an NH2. That's your adenine. Right? And then for guanine, you have NH2 somewhere here. Okay? Right? For guanine, it's an NH2 over here. An extra NH2 amine group over here. Alright? So if you see, it's quite a convenient intermediate. Alright? So at the end of it, if we convert this group, this carbonyl to NH2, then it becomes adenine. And if we add an amino group here, it becomes guanine. Alright? So this is the linking precursor. For synthesis, for purine synthesis, the base is built upon the ribose 5-phosphate. Okay, so the starting material is the ribose 5-phosphate. You will notice it is slightly different from pyrimidine. Pyrimidine is on the other way around. You build the base first. Okay, but over here we start with the phosphate. Okay, we start with the ribose phosphate. So you know this is a ribose sugar, and we number this as 1, carbon 1, 2, 3, 4, 5. Okay, therefore the name is ribose 5-phosphate because the phosphate is found on the 5th carbon over here. Yeah, ribose because it's a 5-membered ring, and this is an alpha configuration. Alpha meaning it is pointing in the plane away. Okay, below. On the 2-dimensional form, we point it downwards, right? So it's a downward projection. The OH ring is on the opposite side of C5 or C6. 1, 2, 3, 4, 5, C5. Okay, OH ring. OH is on the opposite side of C5, right? So we point it downwards. So the first step it will involve the activation of ribose 5-phosphate base. You have, we need to make this very reactive molecule called PRPP. You will see PRPP in pyrimidine biosynthesis, so this step is pretty major. Alright, PRPP stands for phosphoribosal pyrophosphate. So you know when you have 2 phosphate groups together, this is a pyrophosphate. Right? We represent it as PPI. So the pyrophosphate, the ribose 5-phosphate becomes activated when it has lots of phosphate groups added to it. This is the whole basis, right? We study in the basics of metabolism. So over here, okay, so therefore you have the functional group. So if you give this as the main molecule, pyrophosphate, the functional group is a phosphoribosal, okay, 5-phosphoribosal onto the last O, okay, of the phosphate group. So this enzyme, this reaction is catalyzed by the enzyme called ribose phosphate pyrophosphokinases. Right? Again, it's called kinase, right? Because the enzyme adds phosphate group to the substrate. Right? If it's taken out, then we call it a phosphatase. Okay? But because in this case it adds phosphate, so we call it a kinase. So the PRPP. So first now we have to acquire the purine atom N9. Okay, over here. N9 is over here. So we're going to build this 5-membered ring first. Okay, in the purine synthesis, we build this 5-membered imidazole ring first by going from 9. And then we build this 6-membered ring. Okay, so over here, we have to add the purine atom 9. And because this is a high phosphate compound, it has a lot high energy, okay, it's going to be very reactive, right? And we saw the nitrogen is being donated by glutamine itself, right? By glutamine amino acid. Now, when glutamine, you now know the similarity between glutamine and glutamate. Glutamine has an extra NH2. They're very similar, right? So glutamine will donate its nitrogen, right, to the ribose sugar, and in the end becomes a glutamate, leaves the reaction as glutamate, right? And the molecule that is resulted is called a phosphoribosylate, right? Because we have removed the pyrophosphate and added an amine group in that position, and at the same time an inversion occurs, right? Because the final reaction has the sugar, ribose sugar, in the beta position, right? The base is always up here, the sugar at the bottom. So this is the beta position. Previously it's the alpha position. So this enzyme, aminophosphoribosylate transferase, allows the transfer of the amine group and inverts the position. Okay, so the NH2 is now in the correct position in the beta group. Alright, so from phosphate group we build up to the base. Alright, and this compound is very unstable. The half-life is only 30 seconds. Alright, in the body the half-life is only 30 seconds at pH 7. Alright, so that means that it is readily converted. Alright, this reaction moves forward. So the impurine synthesis step 2 is what we call the committed step. Right? It is a no turning back step. Right? Once it moves this way, that's it. The product will be quickly removed, and reaction always goes forward. Right? So that's what we mean by committed step. So once PRA, a highly reactive compound, is made, we now make we build the whole of the 5-membered ring, right? Okay. So we build it up now. Okay, so this is the PRA. In every of the slide, I always put the previous molecule up here. All right. Yeah, I couldn't split it up. So the previous molecule was this, the highly reactive intermediate. Now glycine comes in. Remember we said that we use glycine, whole skeleton of glycine. Glycine is a very simple structure. The R is H. Right, so you have a C, NH3, and then COO minus, and a very simple H group as an R. So you will see that glycine comes in now, and it's being added. Okay, so you have H. Okay, this is the R group. This is the amine group of the amino acid. This is the COOH of the amino acid. So the whole glycine molecule is added to the amino group over here to form an amide bond. Okay, so NH2 plus COOH of the glycine, you form an amide bond. From now onwards, you will see that most of the enzymes are called synthetase. So it's named after the product, which is a glycinamide ribotype. Now you have a ribotype because you have an amide over here, right? So type, T-I-D-E. Okay? And it's called GAR in short, and the enzyme is called GAR synthetase. Right? That's why I say the enzymes is not quite the focus, because most of them are similar, right? In the name, in the action, for example. So over here, the glycine is added to the whole structure. Nothing is removed, wholesale, and it's the only step that has more than one purine ring atom acquired. The rest of the reaction you see very tedious, one atom added step by step, one at a time. But glycine is the most time saving. Okay, add all three together. Okay, add all three and build it up. Okay, so this is the only step with more than one purine ring atom acquired. And once we have the GAR, the glycinamide ribotype, in the next reaction, right, we then add. Okay, we continue to build up this ring. Remember, we have to close up the 5-membered ring. So in this case, we have a carbon group added now. So we already have 1, 2, 3, 4. Four atoms in the 5-membered ring. The last atom is a C. And we saw in the structure, the C is donated by N10 formyl tetrahydrofolate, which is a formyl group, COH group. Right? So in this case, the free amino group, right? We always add on to either the carboxylic group or the amino group. It's easy, it's reactive. Okay, so the free alpha amino group is formylated to form what we call FGAR. So glycinamide ribotype, you just add an F. Formal glycinamide ribotype. And in the end, you have a CHO, aldehyde group. Right? Because the R is an H, therefore it's called a formyl. Okay? Formyl. Right? And the reaction looks like that. I didn't add it in, but I just want to show you how tetrahydrofolate looks like. This is the substrate. It's a bit complicated. You're not required to remember structure, of course, right? But then I want to show you that in this N10, this is number 10. I will send you this slide. N10, CO group, formyl group, yeah, over here, it's been transferred to here, to the amine group, right? And then it becomes just a tetrahydrofolate. The formyl group is removed and becomes just a tetrahydrofolate, right? And then this then undergoes other reaction to regenerate, other enzymatic reaction to regenerate the N10 formyl. Okay, so in case you're wondering where the CO comes from, it's from position number 10 on the nitrogen on the tetrahydrofolate. Okay, right? So our ring is almost complete. We have already 5 atoms on it. What we need to do now with the formal glycinamide ribotype is to close up the ring. Right? To close up the ring. But before we close up the ring, the reaction continues to add another nitrogen. So over here, from here onwards, okay, from here you still have the ribose ring over here, but now after all the reactions are over, we're just going to put the name here, ribose 5-phosphate. Okay, we're going to put just the name here. Yeah? Okay, so you have the FGAR here. Okay, so we will continue to build up the other side now. This is almost complete, right? This is almost complete. So before we close up the ring, let's make another nitrogen on this side. Right? We have to have the lead. The lead is now given by, again, glutamine. So on both sides we saw, right, that the nitrogen is donated by glutamine. Right? Glutamine. So again, let's look for reactions, right? Reactions that we can use for glutamine. Now, glutamine is an NH2. Let's look for a CO or a COOH group. And the only obvious one is this. We cannot use this because we need to close up the ring. So let's reserve this to close up the ring. Now the only available reactive group is over here, the CO. Alright, and in this reaction, the amide group of the second glutamine is transferred to the ring via the CO group, right? And coupled to ATP hydrolysis. And the product is called formal glycinamidine ribotype. Okay? Why? Because over here is a CN double bond now. Right? So from amide, it has changed to amidine. Okay, amide to an amidine. The amide looks like that, CONH. But now, because you have a C double bond NH and NH on the other side, it's now an amidine. Okay? And in this case, the enzyme again is called FGAM synthetase, right? Every time glutamine is added, they call it a synthetase, right? And they name after the product. Okay? So after this step, this will provide the lead to add on to other molecules on the 6-membered ring. And meanwhile, we are able to close up this ring to condense it to what we call an imidazole ring. So imidazole ring is 5-membered with 2 nitrogen here. Okay, with 2 nitrogen here. So we have seen already the imidazole ring has this contributed by glycine, right? And then it's symmetrical. This 2N is from glutamine. Okay, this 2N is from glutamine, and this is from formyl tetrahydrofolate. Okay, right? So it's not difficult to remember. It's symmetrical, right? And over here, the NH group needs to become an NH2, right? CN is a little bit hard to react with. We need an NH2, okay? So over here, the imidazole ring is first closed by a condensation process. So with an NH, we have CO. Right, this condensation process requires ATP and removal. Over here, the ATP is coupled to the reaction, right, to drive it forward. and CO, NH and CO condenses to become a CN group. Okay, condensation process. Right? Okay, so removal of water and then you have condensation process. Right? Condenses, produces water. Like the rain, the water condenses. So you have CN over here, and the ring now is closed up. Okay, and it's called AIR. The aminoimidazole, because imidazole has already been formed. Aminoimidazole ribotide. R is a ribotide. Okay, so the tautomeric shift is from an imine to an enamine. Okay, so what they mean by the tautomeric shift is imine, the C-double-N here, okay, has the double bond has moved up to become C-double-bond-C. Okay, so this becomes an enamine. The imine group has this, over here. C, 2C, double bond N, and then N. But the enamine is when the double bond has moved up here to become CC double bond, and this is an enamine. So tautomeric shift is something like your resonance, right? Your keto tautomeric shift is also like that, moving of the double bonds. Okay. Alright, so now we have the imidazole ring formed. This is kind of left alone already. Okay, so from ribose 5-phosphate, we have built up the one part of the base, the 5-membered ring. And now we are left with this 6-membered ring over here. Right? So it looks like this. The N group, the next one that is being added is the carboxylic group over here at the top, the 6th position. And this purine C6 is introduced as a bicarbonate. Okay, bicarbonate into this double bond. Alright, because this one doesn't have an amine group, it doesn't have a carbonyl group, so we cannot add an amine group in the form of aspartate or glutamine. So it is easier to use the raw form, the bicarb. Alright, the bicarb can be added to a double C bond over here. Alright, okay. In the reaction called carboxylase. Okay, because the COO minus group is added to a carbon group, it's called carboxylation reaction. Right, and the enzyme is a carboxylase. Right? And AIR carboxylase, COO minus from bicarb group. And we mentioned this is the same bicarb, right? Because in Krebs cycle with carbon dioxide being produced, once it dissolves in the liquid or aqueous solution in the cytosol, it becomes a bicarb. And now we have a carboxy, a CAIR, carboxy aminoimidazole ribotide. Okay? So the name gives you a clue of what is being formed in the next reaction. So after CAIR, we have now 2 reactive groups. We have NH2, we have COO minus, right? So it becomes quite nice to add on other materials now. In this case, the next atom that is being formed has to be nitrogen, and an aspartate is being added. Now, the difference between when nitrogen is being added, so we have seen previously in our amino acid lecture, nitrogen atom, if it's not free like ammonia, it can be added to another molecule by a few processes. First one, transamination. Yeah? We pass on the amino group. This is not free ammonia. These are amino group attached already to molecule. We pass it on by transamination. That's the first way. Second way now you have seen, we can use glutamine. Right? So glutamine, in all these reactions, can pass on the amino group to the molecule and becomes glutamine. And the third way to pass on amino group now is aspartate. But the difference with aspartate in this case is that wholesale, the whole molecule is always added to it. Glutamine is just amino group, right? But for aspartate, it's somehow unusual. I don't know why this is this way. Really, I don't know why. But the whole molecule is added. Okay, so aspartate. So you have COO minus NH2, which is your classic amino acid structure, and this is your R group for aspartate. So when it's being added wholesale to the COO group, right? NH2 always look for COO minus for reaction. This is a typical amide bond formation, right? Amide bond formation. So when it's being added, the molecule now becomes quite bulky, right? Very bulky. We don't need everything here, right? And the name here is sidecar, okay? Which now we have the, yeah, doesn't sound very nice, but what it essentially is, it adds on the succinyl appendix, okay? It adds on the succinyl appendix on there. So we have to remove this, right? We have to remove this because all we need is the nitrogen from aspartate. We don't want this. But did you see, this is aspartate, we want nitrogen, and if we remove this, what does it look like? Something we have seen before. Something we have seen before in Krebs cycle, which is the succinate, right? Right, but it is not removed as a succinate. If I add a double bond here, it becomes a fumarate, right? Like Krebs cycle. And also like urea cycle, okay? So in this case, in the next reaction, we have to remove the other carbon skeleton of the aspartate, right? This part here, and it is being removed as a fumarate. A fumarate is COO minus CH double bond CHCO minus. It's a symmetrical molecule. Alright, okay? And this reaction resembles the urea cycle reaction, in which citrulline is aminated to form arginine. Alright, so we mentioned, right, citrulline and arginine, they look very similar in our last lecture. The difference is CO group and the NH2 group. So just to refresh your memory, let me show you the urea cycle. So this is the urea cycle, right? Citrulline is here and arginine is here. Okay, you see citrulline, everything remains the same. The difference is the CO here and then the NH2 here. Okay? Alright, so in this case, in order to make citrulline look like arginine, and aspartate is added wholesale, right? We want a donation of a nitrogen here because we know the conversion is carbonyl to amine. Okay, but they don't choose to have glutamine, but they added the whole aspartate on it. And then you have an intermediate called arginosuccinate. Alright, when this is cleaved, you've got fumarate leaving the urea cycle. And the fumarate is still very useful. Now we saw that it is being used up by other reactions. So fumarate is symmetrical, CO minus, CH double bond, CHCO minus. This is the same reaction. Okay, same reaction. Eliminate the carbon skeleton of aspartate, okay, as a fumarate, you have a double bond here. And in the end, you're left with just an amine group on the CO. Okay, on the CO group. Okay. So now we have 1, 2, 3, 4, 5. We have 5 atoms on the 6-membered ring. We're left with just one more. Okay, we're left with just one more atom. And it's a C. So the final purine atom is a C, is a carbon, right? And this carbon is provided by, again, N10 4-mile tetrahydrofolate. Symmetrical, okay? Symmetrical. Glutamine, glutamine, 4-mile, okay? Aspartate, glycine, bicarb. So this is contributed by the same reaction, okay, called N10 4-mile tetrahydrofolate to the NH2 group. It always looks for NH2 group because you need to add a CO. Alright, so after you add, okay, this reaction to it, the last reaction is to close up the ring. Same. Just now we close up the ring here too. Alright, okay, with condensation reaction. Over here, we can also close it up, right, by eliminating the water. Close it up, okay, by eliminating water. And over here, finally, you have what we want, inosine monophosphate. Okay, the base is inosine with CO on top. This is the skeleton from A and G. Alright, and this is the ribose phosphate group. Okay, this is a ribose group. We haven't, this is only a monophosphate. Right? It's only a monophosphate. Okay? So then IMP is not accumulated in the body. As soon as it is being made, it is being converted to A and G. Right, that's why when you probe for IMP, you rarely see it. Okay? Because what we essentially need in the body is A and G, not I. So this one, the half-life is also very small, low. It never accumulates. Again, so we look at the structure, we don't look at the middle part, let's look at the difference in structure. Inosine, monophosphate, O here, C double O, and adenosine is NH2 over here. Okay, CO, NH2. Right? And this is added to, so directly. And then this one, guanosine, is also an NH2 group. So as I mentioned, there are two ways of adding nitrogen group. Aspartate, or transamination aspartate or glutamine. Right? In this case, for adenosine formation, aspartate is added. And then the same reaction again. You have aspartate. Whenever aspartate is added, it's always wholesale. Alright, it's always wholesale. If I ask you, oh, this molecule is aminated by aspartate reaction, then okay, you must be able to tell me, yeah, aspartate is added wholesale. And you will always have a fumarate production out of it. Okay, fumarate production out of it. So the whole carbon skeleton of aspartate is removed as fumarate. Okay, right? So if I ask you, oh, how can this molecule be aminated? You can give me a few options. Right? You can show me a reaction of transamination. You can show me aspartate reaction. You can show me glutamine reaction. Right? Okay. So over here, the aspartate is added with GTP. Right? And fumarate is removed over here. Finally, we get our AMP. Guanosine, on the other case, will get the intermediate called xanthosine. Xanthosine monophosphate, there's a typo here, it should be xanthosine, not xanthine. Xanthosine has C double O and C double O. So over here, we added a C double O first, right? Because again, troublesome. Amine group reaction, we need a C double O to react with. We cannot just react with C. Okay, the carbon skeleton is highly unreactive, not very reactive, right? So over here, it's better to change it to a C double O or carbonyl. And from carbonyl, we can add glutamine directly, again with the help of ATP or GTP, finally to become GMP. Okay? Alright, okay, so this is, and this one you have seen xanthine in your previous lecture. Dr. Ho gave you an example of gout, of allopurinol. He gave you how hypoxanthine can change to xanthine and then uric acid. Okay, so I'm not going to go through that again. Yeah? He has already taught you on that. But over here, this is the intermediate. Xanthosine monophosphate is the intermediate for GMP. Okay. Okay, so this is purine synthesis. Okay, see how we have already built slowly up, right? Okay, from the aminazole ring first, and then over here, and it's symmetrical. The contribution from different precursors is symmetrical. And this is the structure of pyrimidine. The pyrimidine is C and T. Okay, you remember your pyrimidine? Pyrimidine is only a 6-membered ring, okay? One ring only. Okay, and this is uracil. We have seen this in RNA, right? And this is cytosine. Cytosine has an NH2 over here. See the difference? Okay, this is C. And then finally we have T. Yeah? Okay, so you see from U, if we change CO to NH2, it becomes cytosine. Right? Just now we have inosine, change the NH to become adenosine. And over here, from U, we methylated it. Right? We've got T. Just now it was aminated, right? Then you get G. And then when we talk about transcription, we also say why U is not in DNA, right? Because C can deaminate spontaneously to U. Right? I mentioned this C can deaminate to U. Right? So if we put U in the DNA, we cannot tell whether it is the real C or not the real C. Right? So the message will change and the DNA integrity will change. So that's why nature has it such that you have a methyl group over here to stabilize the structure as compared to U. Okay? So U is less stable. And then U is fine for RNA because RNA is not stable anyway. RNA is constantly produced and removed. So it's okay to have U. In fact, we want to have U, right? But we cannot have it in DNA. We need stability and we cannot have confusing messages, right? Okay? So this is your pyrimidine, right? So again, like the purine, it is C and T is not produced directly, right? Just now A and G is not produced directly. It is through an intermediate called inosine. Over here, we make U first. Okay, for pyrimidines, we make U first. Okay. So again, the key concept is UMP is synthesized first. Right, just now was IMP. But in this case, just now IMP, we build it up from a ribose 5-phosphate. We started with a ribose 5-phosphate, but for pyrimidine, we do it the opposite way. We build the base first. Okay, we build the base, then we add the ribose 5-phosphate. That's a major difference. And from U, we can derive C and U. Yeah, just now from I, we derive a UTP. C and T. T is also derived from UMP. Yeah? Okay? T is also derived from U. Yeah? So just now from I, we derive A and G. Okay, so it's just, you try to group it, organize it laterally. Okay, you can see that the knowledge is quite similar. The concept is similar. And early steps are control points, yeah, but I'm not going to go through regulatory steps. Again, look at the skeleton. Just now we look at skeleton. This is much simpler. Phew, it's much simpler, okay? Over there, just now we have aspartate. Again, that's fine. Aspartate is over here. So in the pyrimidine ring, N is number 1. In this case, the numbering goes clockwise. I don't know what chemists do. Why do they decide sometimes it's anti-clockwise or clockwise, right? It's always one of my worst subjects, medicinal chemistry. Never liked it. I'll tell you all. PR1110. Anyway, so over here, so now it goes clockwise, okay? Yeah? Okay, I like it only when chemistry relates to biology, okay? So over here, N is 1, 2, 3, 4, 5, 6. Yeah? And the carbon, now the whole carbon skeleton on the right side is contributed by aspartate. Okay, it's contributed by aspartate. The red color, all shaded the same color means it's contributed by the same precursor. You can always account for every molecule, every atom, right? And over here, you have glutamine, okay? And here, bicarb. Alright, so let's see how this is being made now, yeah? Again, I put the whole process here so that you can, when you revise, you can have a summary of it. Don't worry about the enzyme pictures on the picture. Uridine. So uridine looks like this. C-double-O, C-double-O. Okay, C-double-O, C-double-O. Uridine monophosphate. Monophosphate ribose sugar. Okay. How do we work it all the way there now? Okay, so over here you have the first thing now. Okay, we have nitrogen. We didn't put, I didn't put this, but N1, first nitrogen is contributed by glutamine. Did I miss out? Yeah, glutamine over here. Oh no, sorry. Aspartate, whole thing. Whole right side is aspartate, red color. A bit hard to see, but glutamine, we build the N on number 3, glutamine first. So only three starting material. Glutamine, bicarb, aspartate, much simpler. Okay, so now carbamoyl phosphate. Does it ring a bell? We have seen this, right? Where have we seen this? Carbamoyl phosphate. So we start with bicarb, glutamine, ATP, catalyzed by the enzyme called carbamoyl phosphate synthetase, but in this case it's number 2. We have seen number 1. Where have we seen number 1 before? Urea cycle, right? Okay, the same product, carbamoyl phosphate. Remember I said that this is a phosphate and this is carbamoyl, CONH2, COONH2. Right? This is a carbamate. COO plus NH2 is a carbamate. With a phosphate group on it is a carbamoyl phosphate. And we have seen this in, right, the amino acid degradation. Okay, so we say that the ammonia, NH3 group from the amino acid, where do we get it from? This is a recall. I pulled it out from the